All right. Good afternoon, each one of you. It's my great pleasure to be part of this scientific forum. I would like to express my thanks to Lachance for inviting me to present some of my research work. I'm going to talk about how the bile acid can improve the performance of the animals and also improves their gut health, especially for farm fish. And I will have some data for shrimp as well. As you know that aquaculture is one of the fastest growing food industry in the world. Um, it doesn't matter if you consider the plant food or animal food. Aquaculture is the fastest food industry, which provides almost 50% of the fish which we consume every day on our plate. Um, it's not the fastest food industry. It is also one of the most uh, efficient food industry in the world. What do you mean? Like if you compare the different types of animal protein food industries, such as beef industry, pork industry, poultry industry, and aquaculture industry. So in terms of beef industry, for example, they need almost 7 kilogram of feed to produce 1 kilogram of beef. In terms of pork industry, you have to provide 4 kilogram of feed to produce 1 kilogram of pork. In poultry industry, you need to provide 2 kilo or more than 2 kilogram of feed to get 1 kilogram of poultry. However, in aquaculture industry, you don't have to provide that amount of feed. In terms of salmonids industry, you just have to provide 1 kilogram of feed to produce 1 kilogram of salmon or trout. Okay, that's why it is considered as one of the most efficient animal food industry in the world. So this is the perfect example to represent why this is the most efficient animal. All right, so aquaculture is growing so fast. So what is their number in another 5 years or 6 years? So according to NOAA, that's called National Atomic Association organization, what we have here in United States. According to NOAA, they did a survey, and according to their survey, by 2030, we need another 40 million tons of seafood on our plate. That means aquaculture has to play important role to provide those additional seafood because our capture fisheries is almost stagnant last 20-30 years. So can we provide another 40 million tons in next 6-7 years? It's almost impossible, but we have to do something to provide it. So what is the bottleneck in aquaculture industry? The major bottleneck in aquaculture industry is the feed. We are struggling every day to find the right feed ingredients to make the enough feed for the aquaculture feed industry. I can give you example here. This picture on the bottom side I took from the Dr. Albert Tekken. He's one of the world-renowned fish nutritionist. According to his review paper, according to his survey, what he found is that last 25 years or 24 years, whatever the feed we predict, and we also, whatever the feed we predict in terms of, let's say, we are predicting by 2020, there will be, according to him, it was, for example, 57 million metric ton of feed we need. By 2025, we need around 69.57 million metric ton of feed. But actually, in real, how much feed we usually use to produce the aquatic animals, they are almost like similar. So what does it mean is that whatever we predict, we are almost there, little bit lower side. So which indicates that every year we need additional amount of feed. Can we provide those feed or not? As I mentioned, ingredient is the major problem. So what is the major ingredient in aquaculture? In aquaculture or in aquafeed industry, soybean is a major feed ingredient. Personally, I do not believe soybean is an alternative ingredient because if you see global aquafeed industry and their composition, on an average, It doesn't matter fin fish and shrimp industry. On an average, soybean contributes on an average around 25% of the total, composition. So, for example, if you have 100% total feed, out of that 25% is the soybean on an average. It doesn't matter which animal you consider in in aquaculture. So that's why personally I believe it is not an alternative ingredients. Okay, it is as a major ingredient. Why soybean is a major ingredient? What is the beauty behind it? Soybean is a great source of oil. You can get those oil, and this oil can be used for human food and animal food, especially even for the fish feed. And it is a great source of pharmaceutical compound, such as there are a lot of pharmaceutical compounds you can get from soybean seed. But the major interest is that when you take out the oil from the seed, that is protein rich meal, that contains almost 40 to 50% protein, and those proteins are well balanced with amino acids except lysine and methionine, which are considered as a limiting amino acid. It is also enriched with vitamins and minerals. Okay, so if everything is great, soybean meal is highly digestible compared to other meals or other plant ingredients. It is a great source of amino acids. So why don't? So what is the problem? There is some problem because soybean meal also contains some of the anti-nutritional factor, which kind of hinders their utilization in carnivore animals such as salmonids, especially trout and salmons. What are those anti-nutritional factor? Trypsin inhibitor and lectin. Excuse me. Trypsin inhibitor and lectin, these are heat level anti-nutritional factors. It's not a problem because most of the soybean meal, what you get it from the market, those are toasted soybean meal. That means those are heated soybean meal. So that means trypsin inhibitor and lectin is not the problem. Phytate, phytate is also not the problem because commercially we have the enzyme available that's called phytase enzyme. You can add those phytase enzyme in the feed, and that could remove those phytic acid easily without any problem because those enzymes can break down those phytic acid, and that will release all those bound phosphorus, and that should be available for animals. Another concern for soybean meal is that it contains a lot of oligosaccharides, almost like 25 to 35% of non-starch polysaccharide and oligosaccharides. That is another problem. However, that's my research area. One of my research areas is that we develop, different processing method to get rid of all those oligosaccharides. So what I do usually in terms of typical nutritional work. So currently on soybean meal, I'm working mostly with the processing and nutritional approach. How can we enhance the soybean inclusion in aquatic feed, aquatic animal feed? So there are different approach. Processing approach, I'm not going to discuss today, but we developed processing technology that could remove the oligosaccharide, and those processed soybean can be used in aquafeed industry. And we have developed one product that could replace 100% fish meal in shrimp industry. So, and that work has been patented already, and we already sold that product. But there are some feed additive approach such as glutamine, bile acid, and vitamin D. These additives can mitigate those gut inflammation in salmonids. As I mentioned earlier, anti-nutritional factor cause some problem in aquatic animals, especially in salmonids. What are those problems? Especially the enteritis. What is the enteritis? In layman word, we can say that enteritis is a gut inflammation in salmonids. And these additives, especially the bile acids, can mitigate those gut inflammation in salmonids. All right. So one of my PhD student is have finished her PhD work, and she graduated and she moved to the Cargill. And what she did is that she used the bile acid, glutamine, and vitamin D to mitigate those enteritis and improve the feed utilization for rainbow trout. And then my another PhD student, she also completed her PhD degree. She's working for Pfizer right now. And what she did is that through the selective breeding program, she was able to identify the animals which are more kind of I mean, like they were able to utilize the soybean more efficiently through the selective breeding program. Okay, that's what she has been doing. She completed her work and she moved to the Pfizer. All right, so let me come back to the soybean meal. As I mentioned earlier, soybean meal causes gut inflammation that is called soybean-induced enteritis. It is mostly happening in the distal intestine. As you know, in the gut, we have proximal intestine and distal intestine, but this inflammation is happening in the distal intestine. On the right side of my slide, this is the perfect example of the how the good health and a good enterocyte and inflamed enterocyte, how they look like. On the top of the picture is the good enterocytes, means there are two enterocytes are joined together by tight junction and they have a finger-like structure microvilli. However, on the bottom side, the picture, they lost the microvilli structure and they lost the tight junction. So the macrophage can enter inside and they can cause inflammation and there are a lot of problems. And what is their cause behind it? What is their mechanism behind the gut inflammation? There is no exact mechanism behind it. However, we hypothesize that this inflammation caused by pro-inflammatory cycles that could lead to increased cytokines expression such as TNF alpha. All right, then it can also lose the barrier function that can ultimately because of the changes in the tight junction proteins. We have the tight junction protein that could break down and that could lead to the leakage of those enterocytes. And then another important protein is called myosin light chain kinase. Okay, that's we have been working on this protein for many, many years, and that could also affect those barrier function. And then all those things will cause the intracellular space that will cause the leakage of the macrophage. And then ultimately it will alter the gut pathology. Okay. However, this is our hypothesis, this is our mechanism. But we do not know the exact mechanism. We are still working on it. All right. So as I mentioned earlier, one of the feed additives which works very well is the bile acids. So what bile acid does, what it is great for improving the gut health. So what do you mean by improving the gut health? What we found is that the bile acids mitigate the gut inflammation, especially in salmonids, especially in trout. Okay, how they do it? Usually they do through the TGR5, which gets activated by the conjugated and unconjugated bile acid. The most potent activator for TGR5 is that it's called TLCA, that is called lithocholic acids, okay? Followed by lithocholic acid, deoxycholic acid, and CDA and cholic acid. There is another method is called bile acid also activate the FXR. And what they do FXR also helps to mitigate the enteritis, okay? So I don't want to go much detail, but the message here is the bile acid activate two pathway, TGR5 and FXR5, that's which we actually, our lab actually come up with this idea in aquatic animals. And based on this idea, we thought, okay, bile acid would be good to add and activate those pathway and mitigate those enteritis. And then we contacted LaChance I think I contacted them in 2019. Yes, 18 or 19. And then we said, hey, can we use your bile acid and see how it could protect gut health and liver health? Because most of the researcher, most of the industry, they were using this bile acid, LaChance bile acid, as an emulsifier. Okay. But we said, no, we can use this bile acid to improve the growth, not just improving the growth, not just as an emulsifier, but also improve the gut health and liver health of the animals. And that's why we come up with this idea. And then we started working with the LaChance. And then here is the typical example that which our lab has designed this typical structure. This picture is from one of our review paper. And how it works. Usually this bile acid gets synthesized in liver, then it goes into the gallbladder. From the gallbladder, it goes into the intestine. And from the intestine, almost 95% of bile acid recycle through the hepatic portal vein, and it goes back to the liver. Only 5% of the bile acid are converted into the secondary bile acid, and then it leaves the intestine through the poop, through the fecal matters. 95% recycles all the time. So if somebody asks you how many times it recycles in the body, in generally 4 to 12 times it recycles. So it's not like it happens every day. It happens once in a day or once in a minute or once in an hour. No, it happens around 4 to 12 times in a day. It depends on the type of the animals. It depends on the nutritional status of the animals, age of the animals, and so on. That's why I put a range is 12 to 4 to 12 times per day it recycles. That's a good thing. All right, and then these things I already mentioned. There are two pathways this bile acid can mitigate the enteritis in aquatic animals. One is the TGR5 and one is the FXR. And we already kind of found that reason. Yes, they do it even in the trout and salmon, okay, especially in trout. So based on those things, we come up with the overall goal of our project was to develop a functional feed additive such as bile acid to mitigate the gut inflammation, okay, as well as reducing the dietary fat and cholesterol in fin, fish, and shrimp. So first objective to achieve this goal was to effects of bile acid to improve the growth performance of the animal, also mitigate the gut inflammation in rainbow trout. And then we come up with the second project where what we did is then in second project, we try to improve the carbohydrate utilization in largemouth bass by reducing the lipid. So when you put the bile acid, what they do the bile acid is that they are, as you know, that it's emulsifier. So if you put more carbohydrate and this high carbohydrate in the body, they make more lipid in the body. And if you have a bile acid, those bile acid would help to mobilize those excess lipid. So overall, you can reduce the lipid inclusion in the diet. Okay, that was our idea, and we were able to do that. And we also look at the liver histology and gut microbiome. And the last project we did was on the can we replace the cholesterol in shrimp diet or not? Answer is yes. Okay, here is the first experiment. I will go quickly. I just want to show you just the summary of our project. In first experiment in rainbow trout, we used the fish meal diet, soybean meal diet, 30% and 40%. And each one of the soybean meal diet, we added that 1.5% bile acid. And we ran this project for 18 weeks, and we sample every 6 weeks, 6 weeks, 12 weeks, and 18 weeks. And we see what happens. And what we found is that it doesn't matter you use the soybean meal, bile acid, or fish meal, there was no significant difference in terms of growth. However, interestingly, after 12 weeks, feed efficiency has been increased, has been, decreased if you use the bile acid. The message from here is that if you use a high amount of soybean meal, such as 40% soybean meal in trout diets, and if you add bile acid in the 40% soybean meal based diet, the feed conversion ratio decreased, which means that bile acid, supplementation of bile acid improve the feed efficiency in rainbow trout. Then we look at the gut histology. Does this bile acid really mitigate the gut enteritis or not? And answer is yes. You could see here on the top, you have the fish meal distal intestine, and then you have soybean meal 30 distal intestine, and you can see clearly that supplementation of bile acid improve the gut inflammation. So this is just the morphological structure, but I want to also show you the cumulative score that matters. What we found in the cumulative score that yes, when you add the bile acid, overall your cumulative score decreased. So if you have a low cumulative score, that means your gut is much healthier. If you have a higher cumulative score, that means you have more vacuolization, more inflammation, more necrosis, more hemorrhage, and more congested vessels. You don't want those things. So bile acid decrease all those numbers, and which is as good as like the fish meal diet. So that means overall message from here is that addition of bile acid or supplementation of bile acid in soybean meal based diet or plant based diets improve the gut health. All right. So concluding remarks from here is functional feed additives such as bile acids did not affect the growth but improve the feed efficiency. So that means it bile acid enhances the feed utilization in rainbow trout, and certainly it mitigates the gut enteritis in rainbow trout. Okay, so I would say that if you are making the feed with a lot of plant protein, especially with the soybean meal diet, I would suggest to add little bit bile acid, because that will mitigate all those deleterious effect which you usually found by the plant diets. Okay, that's the message from this first project. Second project, what we did on the largemouth bass on the different angle. In this case, what we come up with is that our idea is to, can we improve the carbohydrate utilization in largemouth bass by reducing the lipid? Okay, so that was our goal. We want to increase the carbohydrate level and reduce the lipid content in the largemouth bass diet. And if you wanted to see if we add the bile acid, can we utilize, can we enhance the lipid mobilization or not? Okay, first challenge is it's very limited carbohydrate utilization by aquatic animal, especially with trout or largemouth bass. Okay, because if you add more than 20% carbohydrate, then the problem started, especially with omnivore and carnivore animals, by reducing the growth and feed efficiency. Anyway, we come up with that plan, and our goal was that the bile acid will help to mobilize those, lipid utilization. Okay, how? By inhibition of gluconeogenesis. What will they do? This bile acid is that they inhibit the glucose formation, that is called gluconeogenesis. If those carbohydrates will not form the glucose, what will they do? Usually they have to go with another pathway. They make the more lipid, and those lipid will be mobilized by um, bile acid. So here is the typical formulation of the diet. Okay, we have total diets. First diet was high fat diet, low starch diet. So we add 12% fat and 20% lipid. That is called control diet. That's the typical diet for largemouth bass. Okay, so we have three variables: low and high carbohydrate diet, low and high fat diet, and plus and minus bile acid. So total 8 diets. So high fat diet, low starch diet, 12 and 20. High fat, high starch diet, 12 and 30. So we increase the carbohydrate, 10% more carbohydrate in the feed. And then we reduce the fat. You can see 9% fat instead of 12, and we increase the carbohydrate 30. And similarly, low starch, high fat, low starch, high fat, high high starch, but we added the bile acid. So you could see similarly like 4 diets, and those 4 diets were supplemented with bile acid. And what was the answer? Answer here is that I will just come up with a summary. You could see that in terms of weight gain, highest weight gain was with the low fat, high starch diet. And you could see clearly they have the highest growth performance. That means that if you decrease the lipid content, almost like 25% lipid content, if you decreased, and you add more carbohydrate in the diet, but you need to add bile acid, that can give you the best growth. So that way we can make a little bit money in terms of the feed formulation by increasing the carbohydrate source in the diet, because carbohydrate is cheaper ingredient and lipid is expensive ingredient. So we decrease the lipid content, and but bile acid can give you the same result as like the control group. So message is, overall message from here is addition of bile acid can enhance the growth and also enhance the feed efficiency. High diet enriched with carbohydrate and low with fat. So overall cumulative score of distal intestine, you could see clearly was that high fat, high starch has the bad cumulative scores. But if you add the bile acid, okay, they have really great cumulative score compared to the other groups. Okay, you could see here clearly that is the best cumulative score. In terms of liver histology, as we could see clearly that liver histology, low fat, high starch, that was the best. That group has inflammation. You could see clearly granuloma was completely seen. However, if you add the bile acid, those granuloma was mitigated. In terms of gut microbiome, I don't want to go much detail, but what we found was that there was a significant increase in the what's called there was no significant effects between the diets with the fat and starch. However, a significant increase was observed by the addition of bile acid, especially with the high fat, low starch diet. Okay. In terms of, we'll also look the beta diversity. In terms of beta diversity, what we found is that there was a significant clustering because of the bile addition. That means that helps those microbes to be kind of maintaining their structure or maintaining their integrity. And then we also look at the relative abundance, okay? And then in terms of the microbial composition between the dietary, between the different treatments, you could see clearly here that the relative abundance of the most abundant genera in the digesta, as you know, and there was a significant difference. And what we found is that one of the most was that there was a not significant difference among those relative abundance actually. So overall conclusion here is that bile acid seems to have a positive effect in terms of growth and liver histology. Okay, and gut microbiome was affected, but because of I had limited time, so I could not able to explain you much about the gut microbiome. But I do have more information about the microbiome, that how the bile acid affects the gut microbiome, and in a positive way. Okay, so overall message from this project was the bile acid has a positive effects. Okay, in this one. Last slide of my presentation is that we also run some project, or we are running some project in shrimp industry. And as you know, that shrimp cannot synthesize cholesterol. Okay, sorry, cannot synthesize bile acid. Okay, they have to get it through the feed. Bile acid, what they do is that they promote the lipid metabolism, and in general, shrimp also cannot synthesize cholesterol, so you have to add cholesterol in the feed. So what happened is that cholesterol is very, very expensive. It could be almost $100 per kilo. So we come up with the idea: can we reduce the cholesterol level in the shrimp diet? Answer is yes. You could reduce cholesterol up to 100%. But personally, I would recommend that you should use cholesterol 50-50. Okay, you can replace 50% cholesterol by bile acid with no problem. So uh, scientifically, 100% cholesterol can be replaced by bile acid, but personally, I would recommend to mix cholesterol and bile acid for the shrimp feed industry. Take home message is the functional feed additives such as bile acid can improve the feed efficiency and also improve the gut health for trout. And then in terms of largemouth bass industry, you can use more carbohydrate and reduce the fat level in the diet, but you should add the bile acid in the diet. Okay. Cholesterol can be replaced by bile acid in shrimp feed industry. As I mentioned earlier, I would recommend 50-50. You should use 50% bile acid and 50% cholesterol. That will be great for the shrimp feed industry. So I would recommend each one of you do your health a favor, eat seafood. And I believe personally that sustainable aquaculture is the better source for feeding the future. All right. I would like to acknowledge my lab and different source of funding. If you have any question, here is my information. I would be happy to answer any question. Okay.